Associate Professor of Anatomical and Surgical Pathology in Tehran University of Medical Science. We want to speak about, can you hear me? I should speak louder. Can you hear me? And can you see the page in full screen?
Yeah.
My head is not in front of you.
No problem. It's good.
And how many students you should be?
Yeah, I have just. Can you wait for 5 minutes?
Yes. We want to speak about disease of immune system. It's season 5 from Pathology Robbins book, and we made this season in 4 sessions, 2 sessions this week and 2 other sessions in another. Okay. So we first want to speak about basic of immunology. First, we want to speak about immunity. What is immunity? Immunity refers to protection against infection. The immune system is a collection of cells and molecules that are responsible for defending body against the external pathogens or some of bacteria, fungal elements, protozoa, or many infections. Okay. Defect in immune system render individual easy prey to infectious and are causes of immunodeficiency disease. Okay? But immune system is itself capable of causing tissue injury and disease, which we can see in hypersensitivity disorder or autoimmune disorder. Okay, when immune system is activated more than needed in our body, we have hypersensitivity disorders or autoimmune disorders. Defense against pathogens consists of 2 types of reaction. We have 2 types of immunity. Innate immunity, which also called natural immunity or native immunity, is mediated by cells and proteins that are always present. Okay? We have also adaptive immunity. What is adaptive immunity? It's normally silent in our body, and when we encounter an infectious process, the adaptive immunity is activated and generating potent mechanisms for neutralizing and eliminating the pathogen. So the innate immunity is always present in our body, and adaptive immunity is activated when we encounter the pathogen. Okay. Do you know? Can you name some parts of innate immunity?
Neutrophil, macrophage, barriers.
Yes, skin, epithelial tissue, macrophages, natural killer cells, yes.
Phagocytes.
Yes, phagocytes. These are the, yes. And can you name some part of adaptive immunity?
T cells,
Yes, yes. They are, yes, lymphocytes are the major part of adaptive immunity. Innate immunity calls into action and immediately in response to infections, provide the first line of defense. A major reaction of innate immunity is inflammation. We speak about inflammation in other season of Pathology Robbins. In season 3, we have speak in detail about acute and chronic inflammation. And innate immunity usually lead to an inflammatory process. Adaptive immunity is normally silent, and by convention, the terms immune system and immune response generally refer to adaptive immunity. Okay, adaptive immunity is maybe more important in immunologic disease more than innate immunity in immune disorders. Okay. These are the major components of innate immunity as you tell. Epithelial barriers of skin, gastrointestinal tract, respiratory tract, phagocytic leukocytes such as neutrophils and macrophages, natural killer cells, and several circulating plasma proteins, such as complement system. Complement system. And we can you see the pointer? Or I should use it.
Ma'am, why complement system is placed in innate immunity as it needs antibody to activate?
Because in complement is produced by the other component of innate immunity, and it may help in neutralizing or opsonization of the bacteria, and it's always present in our body, not in when we encounter a new pathogen. Complement is always active in our body, and adaptive immunity is basically based on lymphocytes, and lymphocytes, T lymphocytes, production of antibody or cytotoxic effect of T cells. Adaptive immunity is based on these steps, but complement is so a part of innate immunity because it's always present in our body. We do not need to encounter a new pathogen or complement to be activated.
For activation of complement, we need either pathogen or antibody.
Yes.
It's not in activated form in our body. Innate cells are already active in our body, but for activation of complement system, we need antibody.
It's present, but when it will be more activated when we encounter the new pathogen. But it's present in the basic default. Okay. Here we have innate immunity and adaptive immunity. It's epithelial barrier here, phagocytes here, dendritic cells here, natural killer cells, and complement system here. We have time of infection. In the first hour of the infection, usually innate immunity have the major part, and when the time go past, from 1 day to 5 days from the beginning of the infectious process, adaptive immunity have the major part. So innate immunity we have only in first hours of the infectious process, and adaptive immunity are composed of B lymphocytes here, and they turn to what B lymphocyte turn to? What are these? Do you know what are these? Colonality, or what is this cell? I mean.
Plasma B cells.
Plasma cells? Yes, plasma cells. B lymphocytes turn into plasma cells when they activated. We speak about this process in detail in other slides. But these are plasma cells which produce antibodies, and antibodies are against the external pathogens. And here we have T lymphocytes, which T lymphocytes need. Do you know what is this cell? Yes, antigen presenting cell. Antigen presenting cell present the antigen to T lymphocytes, and they turn to some maybe they may be CD8 positive T lymphocytes or CD4 positive T lymphocytes, or we have some memory cells, and the pathogen will be eliminated. We speak about the innate adaptive immunity in detail in other slides.
In this slide, how complement system activate in first stage of complement system? We have three pathways. Alternative pathway, classical pathway, or Manos binding victim pathway. For alternative pathway, classical pathway, activation of these pathways, we need antibody. An antibody produced after 1 or 2 days. Then how complement system activate in first 12 hours? For the activation of complement system, we need antibodies.
Okay, we need antibodies. I get you. You mean for activation of complement system, you need antibodies. Okay, so you want to relation between adaptive immunity and innate immunity.
And it must be the complement system must activate after B cell activation.
Yes.
Because we need antibody.
Yes, we do not need antibody in all of the pathways of complement activation.
But it's specific for some.
Yes, I know. You mean we need antibodies for some of pathways of complement system. So you want to test it's the relation between adaptive immunity and innate immunity. They can be related to each other, but complement in all of the pathways will not need antibody for activation. Maybe in some of the pathways you need antibody, but not in all of the pathways activation. You know. Get your question. Okay, innate immunity. Phagocytes, dendritic cell, and many other cells, such as epithelial cell, express receptor that sense the presence of infectious agents and substances released from dead cells. Receptors of innate immunity. It's very important. We want to speak about receptors of innate immunity. We have some microbial structures recognized by receptors that called pathogen-associated molecular pattern. We have pathogen-associated molecular pattern. These are some receptors that are sensitive to some molecules of the bacteria or fungal elements. Okay, and the substance released from injured and necrotic cells are called damage-associated molecular pattern. So we have two molecular patterns, pathogen-associated molecular pattern and damage-associated molecular pattern. The cellular receptors that recognize these molecules are called pattern recognition receptors. Pattern recognition receptors are located in all the cellular compartments where pathogen may be present. They may be present in plasma membrane as plasma membrane receptors, which detect extracellular bacteria or extracellular pathogens. We have endosomal receptor, detect ingested microbes, and we have cytosolic receptor, detect microbes in the cytoplasm. So the receptor may be in all of the cell compartments.
These are type of PRR. These receptors are.
What?
These receptors are type of PRR receptors.
B cell receptors.
PRR. Receptors. Can you?
We speak about all of these receptors. Okay, we have three types of receptors. We have some, and here you can see they are in plasma membrane receptor here, and we have endosomal receptor here, and cytosolic receptor. The type of the receptor are different. For example, we have TLR or toll-like receptors in the plasma membrane, and we have some others such as NOD-like receptor in the cytosol, and we have some endosomal receptor. We speak about all of these receptors.
We mentioned are the types of PRR receptor, pattern recognition receptor, or there are some different receptors.
And the toll-like receptor are pathogen-associated receptor, and for example, NOD-like receptor are damage-associated receptor. Yes. Okay, we can see this is extracellular matrix here, and this is plasma membrane here, and we have a toll-like receptor here, and a toll-like receptor can design pathogen recognition pattern receptors, which recognize bacterial product here. Okay, and we have some lectin receptor, which is sensitive to microbial polysaccharides, such as it's very active in fungal infection, for example. And here we have NOD-like receptor, which may be sensitive to bacterial peptidoglycan or products of damaged cells. So this is a damage-associated pattern receptor. Okay, so to NOD-like receptor, for example. And we have
For the complementary system, the lectin one.
yes, monoclonal lectin. You mean monoclonal lectin? Yes, it's one of the pathways of complement system. Yes. And this is regular receptor here, which is sensitive to viral RNA here. And we have some endosomal receptor here, which is sensitive to nucleic acid of ingested microbes. It has an endosomal membrane okay, in the endosomal membrane. So receptors of innate immunity are toll-like receptors, node-like receptors, and we have some other receptors for microbial products. The major receptors are toll-like receptor and node-like receptors. Toll-like receptor, the best known of pattern recognition receptor. We have 10 toll-like receptors in mammals. The plasma membrane toll-like receptors recognize bacterial products such as lipopolysaccharide. And we have also endosomal toll-like receptor which recognize viral and bacterial RNA and DNA. You can see in here, we have toll-like receptor endosomal in endosomal form here, and we have in plasma membrane form here, toll-like receptor. That was endosomal and plasma membrane.
Ma'am, toll-like receptor is specific for only bacteria.
No, not only bacteria. It's sensitive to, for example, bacteria, but other pathogen can also activate the toll-like receptor. Not only bacteria. When toll-like receptor are activated, it causes stimulation of production of mediators of inflammation such as antiviral, cytokines, interferons, and promote lymphocyte activation. Okay, it produces antiviral cytokines, so it's sensitive. Here you can see it produces antiviral cytokines, so it's sensitive also to viral proteins as well as bacterial proteins. And we have node-like receptors and inflammasome. Node-like receptors are cytosolic receptors which recognize products of necrotic cells such as uric acid or release ATP. When cells is injured, ATP released from injury of mitochondria. Okay, so the level of ATP in the cells elevated. Okay, or maybe uric acid or lung disturbance to loss of potassium and some microbial products. Why we have a loss of potassium in injured cells? Why the level of potassium decreased in the injured cells?
Due to less ATP, due to the less number of ATP, because sodium potassium pump is not
Will be
working.
not
Due to a proton pump inhibitor in the plasma membrane. We have some ion pump, okay, in the plasma membrane, and when a plasma membrane is injured, for example, in the necrosis or apoptosis, mainly in necrosis, the plasma membrane is disturbed, and the pump in the plasma membrane are injured, and potassium go out of the cells. For example, due to sodium-potassium pump in the plasma membrane, to injure to sodium-potassium pump in the cell membrane. So when this happens, loss of potassium, uric acid accumulation, and elevation of ATP, these processes activate node-like receptors in the cytoplasm. And we have a complex named inflammasome. I show you the picture of inflammasome. Inflammasome is a cytosolic multiprotein complex which activates enzyme caspase-1 and lead to activation of interleukin-1. Interleukin, what's the function of interleukin-1? Interleukin-1, yes, requires leukocyte and induce fever. Fever is very important in activation of interleukin-1. Gain of function mutation in one of node-like receptor results in periodic fever syndrome called autoinflammatory syndrome. So we have a connection between inflammasome and node-like receptors. When node-like receptors are overactivated, we have overactivation of inflammasome. And inflammasome lead to production of interleukin 1, and interleukin 1 elevation lead to periodic fevers, autoinflammatory syndrome. Okay. This is the
Then this inflammasome is activated by node-like receptors.
node-like receptors, yes. Activated by node-like receptor, but not tolerant. For
These
example,
are these inflammasomes are not receptors.
inflammasome activated but not-like receptors in the cytosol.
Cytosol.
They do not activate by toll-like receptor or ring-like receptor, other receptors. You can see here, it's the picture from your book. You can see here, and this is you can hear bacterial products, for example, or crystals, for example, in gout disease, accumulation of uric acid, and we have potassium flux or presence of reactive oxygen species here. They lead to activation of node-like NLRP means node-like receptor. Okay, it's abbreviation of node-like receptors, and node-like receptors are activated here, and it's lead to activation of we have here inactive caspase 1. And this is the inflammasome complex here. Inflammasome complex activate the caspase 1, this inactivate caspase 1 here, and we have active caspase 1 here, and it's lead to production of interleukin 1. Okay. So inflammasome are activated by node-like receptors. Node-like receptors may be activated by crystals, potassium flux, or it can be also activated by bacterial products. Okay. Because we have some disease, pre-disease named here that are in connection with activation of node-like receptor and inflammasome. For example, recognition of urate crystal by a class of node-like receptors lead to inflammation associated with gut disease. And we have node-like receptor respond to lipids and cholesterol crystals, resulting inflammatory may contribute to obesity-associated type 2 diabetes and atherosclerosis. So we have three diseases which are in connection with activation of inflammasome. Gut disease, atherosclerosis, and obesity-associated type 2 diabetes. We have it's very common to be questioned in the exams. What disease is associated with activation of inflammasome in your exam? They may be questioned. Okay. We have
Then,
a small.
can you explain the last slide again? This code, can you explain it again?
Well,
These three diseases, can you explain it again?
explain what the disease. In gut disease, we have accumulation of uric acid, okay? And we speak about when we have urate crystals. Here we have the urate crystals, okay? Crystals here are predominantly urate crystals. And urate crystals lead to activation of node-like receptor and inflammasome. And in clinic, there are some people which they have high level of uric acid or BUN or urate in the serum, in the serum, and they are very prone to bone pains or some of the edema and in the joints, edema in the joints, especially in the lower part of the body, for example, in foot or in the in some joints in the foot.
Then in this disease, this node-like receptor is recognized in the crystals,
Yes, yes,
and this is an immune response.
yes.
So what is the disease?
What is the disease? It's gut. The name of the disease is gut.
This is an immune response
Yes, yes.
to remove the crystals. What is the disease in this?
It's when the NOD-like receptor activates the inflammasome, it's linked to inflammation. It can be a focal inflammation, and focal inflammation lead to bone pain and joint pain. It's due to inflammation. It's an auto-inflammatory process. Uracil cells activate the pathway, and inflammation, which is local in the joints, makes the symptoms of patients. Okay.
And in the case of cholesterol,
In the cholesterol, it's first we have some cholesterol plaque, but when the NOD-like receptors are activated, inflammation is activated, the inflammation is the next step of the inflammation activation. And when inflammation is activated, we have the plaque we have the signs of inflammation, edema, congestion, presence of many inflammatory cells, and it's lead to symptoms of the patient. Okay, in autorespiratory.
in all these diseases there are inflammation.
Well,
In all of
yes,
these
yes, all of the symptoms are due to activation of inflammation. In season 3, activation of inflammation makes the patient see symptoms. Okay. We have so the major receptors here are toll-like receptor and NOD-like receptor. Okay, but we have other receptors for microbial products such as C-type lectin receptors or CLRs expressed in the plasma membrane, macrophages and dendritic cells which detect fungal glycans. I'll show you the receptors in the first picture. So we have also several cytosolic receptors detect the nucleic acid of viruses, for example, RIG-like receptor, or IG or RIG-like receptors detect the nucleic acid of viruses. We have some other receptors such as G protein coupled receptors which recognize short bacterial peptides containing N-formyl methionine residue. The name of this receptor are named in the text of Robbins, but we do not, the Robbins do not show any picture from these receptors. Maybe due to they are rarely activated in comparison to toll-like receptor or NOD-like receptor, but these are named in the Robbins text. And all bacterial proteins and few mammalian proteins, only those sensitized within mitochondria, are initiated by N-formyl methionine stimulate chemotactic response. Okay, so the activation of G protein coupled receptor lead to chemotactic response in the phagocytes. Monos receptors recognize microbial sugars. For example, we have some bacterial sugars, terminal monos residue, which induce phagocytosis. And we have RIG-1 and other called cytosolic DNA sensors recognize microbial RNA and DNA. Okay, so we speak about the receptors. So, when receptors are activated, we have inflammation. And inflammation leads to production of cytokines and product of complement activation, trigger the vascular and cellular component of inflammation. For example, in antiviral defense, type 1 interferon produced in response to viruses and activate enzymes that degrade viral nucleic acid and inhibit viral replication. Which was the viral receptors, antiviral receptors?
NOD-like
RIG-like receptors. For example, RIG-like receptors are activated, and then the activation of RIG-like receptor leads to production of interferon from macrophages, and you have some enzyme activation which degrade viral nucleic acids. Now we want to speak about adaptive immunity. Adaptive immunity consists of lymphocytes and their products, including antibodies. There are two types of adaptive immunity: humoral immunity and cellular immunity. Humoral immunity mediated by soluble proteins called antibodies that are produced by B lymphocytes and protecting against extracellular pathogens. Okay, so humoral immunity, B lymphocytes, extracellular pathogens. We have also cellular immunity, which is mediated by T lymphocytes and defense against intracellular microbes. Okay. And this is a comparison between innate vs adaptive immunity. In innate immunity, we have epithelial barrier, phagocytes, complement system, natural killer cells, and in adaptive immunity, we have humoral and cellular immunity. Let's talk about this. And this is a comparison between humoral immunity and cellular immunity. Can you see here? We have some extracellular microbes here, and we have B lymphocytes. B lymphocytes have antibody in their surfaces, and B lymphocytes recognize the extracellular microbes, and it may have production of antibody. Maybe go to passive neutralization of the microbes or opsonization of the microbes for phagocytes. Okay? And we have cellular immunity, which is sensitive to intracellular microbes such as viruses. Bacteria is more extracellular microbes, and viruses are intracellular microbes. So humoral immunity is more against bacterial infection, but cellular immunity is against viruses, for example, intracellular infection. Yes.
You said cellular immunity is like intracellular immunity.
Yes, yes.
In the cellular immunity, like we have CD4 receptor. It's for the extracellular pathogen. So how it can be like intracellular immunity?
Yes. I will show you this. We speak about T cell receptor and CD4, which is it's in the connection with antigen presenting cell, and I will show you the CD4 part.
How we can differentiate the extracellular and intracellular pathogens? On what basis we categorize the pathogens in the intracellular?
Yes, the book name bacterial elements, maybe due to the size of them, they are extracellular, maybe due to their size. But viral elements or viruses may be due to their size or the silent presence of viruses. They may come in intracellular. But bacteria are in the size large, and they can be silent in our body. We do not have any intracellular bacteria, but we have some intracellular viruses in our body because maybe due to their size or maybe due to a silent nature of viral infection.
But we have some intracellular bacteria also.
Like symbiotic.
Immediate what?
Some bacteria also in intracellular.
Yes, you mean some bacteria may be intracellular or they may be extra. It's a bit speak about majority of the bacteria. Yes, maybe we have some exception. Maybe we may have some exception, but totally the bacteria, majority of bacteria are extracellular. We may have some intracellular in exception. Okay, but we speak about majority of the infections here.
We can determine it by MHC. Can we determine it by the MHC if it attacks
MHC.
to
Yes, I speak about MHC activation in other slides in detail. Okay. See the other slides, and if you have the question after the slides, I will answer the questions.
When to activate B cells, we don't need any antigen presenting cells.
Yes.
So why it is late? Why it activates? It takes time to activate. It must be activated on the same time like innate immunity. Why it takes time?
Because in innate immunity, it's always present. The major difference between innate immunity and adaptive immunity is activation of adaptive immunity. We should have antigen encounters in the adaptive immunity, but in innate immunity it's always present. We do not any encounters to antigen. Okay. The major difference between innate and adaptive is the need to antigen to be encountered the B lymphocytes by antigen presenting cell or not. Okay, but in innate immunity, we always have barriers. We always have epithelial barriers. We always have phagocytes. We do not need to antigen to be encountered to our body. Okay, the major difference is here. The difference is not in presented by antigen presenting to antigen presenting cell. Okay.
It means this B cells is activated.
I speak about I speak about B cell activation, T cell activation, please wait, and if you have question after the explanation, you will answer the question. Okay. Let me speak about antigen presenting cell, and if you have question, I will answer. Okay, cells and tissues of immune system. We have lymphocytes. Lymphocytes present in circulation and various lymphoid organs. Lymphocytes develop from precursors in generative lymphoid organs. T lymphocyte mature in thymus, whereas B lymphocyte mature in bone marrow. A process unique to lymphocyte differentiation is the rearrangement of the gene encoding antigen, a specific receptor on B and T cells. It's very interesting. Every B cells have a specific antibody, a specific receptor for a specific antigen on the surface, and it's due to a process named gene rearrangement in the maturation of the lymphocytes. Each T or B lymphocyte and its progeny, which constitute a clone, we name it clone, express a single antigen receptor, and the total population of lymphocytes, numbering about 10-12 in humans, can recognize 10 or hundreds of millions of antigen. So we have millions of antigen and we have millions of lymphocytes in our body, which is specific to each antigen. The enormous diversity of antigen receptors is encoded by variant DNA sequences that are created during lymphocyte maturation. We have B lymphocyte here, and this is naive lymphocytes. Naive B lymphocytes here, and they have some antibody or receptors on the surface of naive lymphocytes. And the lymphocyte, B lymphocyte encountered a microbe, and the maturation of B lymphocyte completed here. They turn to plasma cells and production of antibodies. Antibodies activate the complement system. One of you asked about complement system. Yes, we have activation of complement system by production of antibody here. But complement system can be activated by other methods in innate immunity. Complement have a role in both adaptive immunity and innate immunity. And function of antibody are neutralization of microbes, phagocytosis, and complement activation. The antigen receptors that are expressed in B cells are called antibody, while their T cell counterparts are called T cell receptors. Mature T and B lymphocytes recirculate through peripheral or secondary lymphoid organs, for example, lymph nodes, spleen, and mucosal tissue. So, where was the primary lymphoid organs? It was bone marrow for B cells and thymus for T cells. But we have peripheral or secondary organs such as lymph nodes, spleen, and mucosal tissue. We have a process called clonal selection. foreign antigens that are concentrated in these organs, in peripheral lymphoid organs, where they bind and activate the clones of lymphocytes that express receptor for those antigens. Okay, so the specificity of lymphocytes for any antigen happen in the secondary lymphoid organs, which the pathogens or antigens are concentrated there, and the lymphocytes are the lymphocytes encountered the antigens in the secondary lymphoid organs, and they make some gene rearrangement to be specific to that antigen. Okay. What are the phases of lymphocyte maturation? We have named naive lymphocytes. Naive lymphocytes express antigen receptors but have not responded to antigen and do not serve any function. Naive lymphocytes have not been met any antigen. Okay? Effector lymphocytes are induced by lymphocyte activation and perform the function that eliminate the microbes. And we have memory lymphocytes. Memory lymphocytes are activated and survive in silent state after the antigen elimination and respond rapidly upon the subsequent encounters. Okay, so we have innate lymphocytes, we have effector lymphocytes, and memory lymphocytes. T lymphocytes, we have two or three types of T lymphocytes. We have T helper or CD4 positive cells, which help or stimulate B cells to produce antibodies. Okay, and macrophages to destroy the ingested microbes.
The memory lymphocytes, they are usually related to the B cells or T cells.
What?
The memory lymphocytes, can they be also included in the T cells? They are only the B cells.
B cells. Yes, we're having B cell activation. And we have T cytotoxic CD8 positive cells, which recognize and kill virus-infected or tumorigenic host cells. T cells constitute about 60 to 70% of lymphocyte in peripheral blood, and they present in splenic, periarteriolar sheets, and lymph node interfollicular zone. Okay? So the major part which we can find T lymphocytes are splenic, periarteriolar sheets, and lymph node interfollicular zone. Okay. In activation of T lymphocytes, we need antigen-presenting cells which capture, process, and present microbial or non-self antigens and present them to T cells. T cells only recognize antigen presented by other cells, so we need antigen presenting for activation of T cells. Okay? The antigen presenting cell or accessory cells is the cell that displays antigen bound by major MHC or histocompatibility complex protein on its surface. Okay, T cell receptor. T cell receptor, when the peptide antigens presented by MHC molecules are recognized by TCR T cell receptors. The binding between T cell receptor and antigen peptide is of relatively low affinity, and it may be degenerated. The T cell receptor is composed of two different protein chains, alpha chain and beta chain in 95%, but may have some gamma delta chain in 5%. Do you know where the T cells which have gamma delta chains present in our body? What?
The delta and gamma,
Delta gamma.
they are regulated.
Regulatory, no. They present in mucosal surface. They present majorly in the mucosal surface of our body. So So the majority of our T cells, 95% of our T cells in our body are alpha beta chain. And we have rarely gamma delta T cells which present in mucosal barrier in our body. Each chain, alpha beta chain, has a variable region that participates in binding a particular peptide antigen. When TCR engages with antigenic peptide and MHC peptide complex maybe here, the T lymphocyte is activated through signal transduction. I will show you this in the picture. Alpha and beta chains expressed as part of complex with invariant CD3 chain molecules. Each chain is composed of two extracellular domains, means variable region and a constant C region. The constant region is proximal to cell membrane, followed by a transmembrane region that is short cytoplasmic page, while the variable region bonds to peptide MHC complex. I will show you this picture here. This is the picture of T cell receptor. Here we have antigen presenting cell, class 2 MHC molecule here, okay, and this is T cell. This is a T cell. For example, it's a CD4 positive T cell, but it may be a CD8 positive T cell, but if we have CD8 positive T cell, we have MHC class 1 here. But the example is about CD4 positive T cell and class 2 MHC here. And this is alpha and beta chain. And we have a CD3 complex here, CD3 complex here, CD3 complex here. And we have some B chain here, okay, and we have CD4 and CD28 complex here. CD4 and CD28 complex they are co-stimulator. They need to be present in activation of T cell receptors. And we have alpha and beta chain here. And we have CD3 complex and Mu chain here. CD4 and CD28 are also involved in T cell activation. Note that some T cell express CD8 and some of them express CD4. We have it shown CD4 here. And this is the MHC complex. So MHC complex and antigen presenting cell present the peptide antigen here to T cells. And these are the, we have some variable part and we have constant part. This is variable region of the T cell receptor. Are you tired?
Where?
And these are the types of T lymphocytes. We have T helper
Where?
cells.
Can you show the last slide? The delta and gamma chain are just
Delta gamma.
this for structural support. These two
Yes,
chains.
delta gamma.
Yes.
Delta gamma maybe we have it show alpha beta, but we may have delta gamma. Delta gamma T cells do not need antigen presenting, and they activate faster than alpha beta T cells, but they are rare in our body. Only 5% of T cells in our body.
And what is function of this CD3?
CD3. Yes, CD3. It may have CD3. What's the function of CD3? CD3 complex is needed for activation of the T cells and all of the T cells. For example, CD4 positive or CD8 positive, most of them have CD3 in their surface, and it needs for activation of T cell receptor. We have T helper lymphocytes here, and we have three types of T lymphocytes in our body. Here we have T helper lymphocytes, and we have cytotoxic T lymphocytes and regulatory T lymphocytes. T T helper lymphocytes are activated by microbial antigen presenting by antigen presenting cell here, and the major function of T helper lymphocytes are production of cytokines. Okay, and when the cytokines are produced, we have activation of macrophages, we have inflammatory response, and we have activation, proliferation, and differentiation of both B cells and T cells. T helper lymphocytes. Here we have T cytotoxic lymphocytes, CD8 positive, this is CD4 positive, and we have infected cells presenting antigen, microbial antigen, and here we have activation of cytotoxic T lymphocytes, and cytotoxic lymphocyte function is killing of the infected cells. And we have regulatory T lymphocytes. Regulatory T lymphocytes lead to, the major function is suppression of immune response. And regulatory T lymphocytes are needed for subsiding, for suppression of the immune response. They are anti-inflammatory cells. Okay, we have CD4 and CD8 on the T cells, and we have CD28 which functions as the receptor for molecules called co-stimulators. CD28 is a co-stimulator that are induced on APC by microbes. CD4 molecules on T cells bind to invariant portion of class 2 MHC molecules, which expressed in 50 to 60% of T cells, and CD8 positive T cells bind to class 1 MHC molecules, which expressed on above 40% of T cells. So we most of our T cells in our body are CD4 positive. The number of CD4 positive are more than CD8 positive in our body. CD4 positive T cells are called helper T cells that help B cells to produce antibodies and macrophages to destroy phagocyted microbes. And we have HIV infection or HIV virus destruct CD4 positive helper cells. And the most important role of CD8 positive T cells is to directly kill virus infected cells. And we have regulatory T lymphocytes which suppress immune response, gamma delta T cells which do not express CD4 or CD8, recognize non-protein molecules, for example, bacterial lipoproteins. Okay? So gamma delta T cells do not have CD4 or CD8. And we have also another small population of T cells which express the marker of T cells and natural killer cells. We name these natural killer T cells, which recognize microbial glycopeptide, the glycolipid. The antigen receptor of gamma delta T cells and natural killer T cells recognize antigen independently of MHC molecule. Okay? So, for gamma delta T cells and for natural killer T cells, they do not have any, they do not need any MHC molecule for activation.
Ma'am, this gamma delta T cells don't have any CD4 or CD8. This gamma delta T cells is not CD8.
Gamma delta T cells,
Yes.
they do not have any CD8, they do not have any CD4, they only have CD3 complex in their receptors, and they do not meet any antigen presenting, and they are in the mucosal surface, and it's very in association with innate immunity, for example, because they do not have any.
They are inactivated form or activated form, like they can perform its function, or they have to be converted into CD4 or CD8.
what's the function of them?
Yes.
You mean the function of them? They have some do not the function of them is not like CD4. It's more like CD8 positive. They have cytotoxic effect, but they are in the mucosal barrier and they do not any antigen presenting. Okay. CD4 positive or T helper cells, they produce cytokines. Okay. But gamma delta T cells are more in function, but more like in CD8 positive, but they do not need any antigen presenting. They are very low concentration in our body and only in the mucosal surfaces. But we have some types of lymphoma. For example, gamma delta T cell lymphoma. We have they make the process of lymphoma, but they are very rare. It's a very rare type of lymphoma, gamma delta T cell lymphoma. Okay. Now we want to speak about major histocompatibility complex molecules. MHC. The normal function of MHC molecules is to display peptide for recognition by CD4 positive and CD8 positive T lymphocytes. In each person, T cell recognize only peptides displayed by that person's MHC molecules, which of course are the only MHC molecules that the T cells normally encounter. This phenomenon is called MHC restriction. Okay? And we have HLA, the human MHC, known as the human leukocyte antigen complex, consists of cluster of genes on chromosome 6. So HLA are the genes in the chromosomes, and MHC are the proteins which are in the surface of the cells. MHC gene products fall into two main categories: class 1 MHC, class 2 MHC. Class 1 MHC molecules are expressed on all nucleated cells and are encoded by three closely linked loci designated HLA-A, HLA-B, and HLA-C. Each of these molecules consists of a polymorphic, an alpha chain, non-covalently associated with an invariant beta-2 microglobulin polypeptide. Okay, so in MHC1 molecules, we have a polymorphic alpha chain and a beta microglobulin polypeptide. Foreign peptide bind to extracellular portion of alpha chain. There is also a considered region that binds CD8. Class 1 MHC molecules bind and display peptides derived from protein antigens present in the cytosol of the cells, for example, viral or tumor antigens. We have also class 2 MHC molecules that are encoded by genes in HLA-D region, which contains three sub-regions: HLA-DP, DQ, and DR. It expresses mainly on antigen-presenting cells. So we have class 2 MHC molecule antigen presenting cell, but class 1 MHC all of nucleated cells in our body. Notably dendritic cells, macrophages, and T cells. It responds to extracellular proteins such as microbes that are ingested. HLA genes are highly polymorphic. Each individual expresses only one set of HLA genes. Each set of maternal and paternal HLA genes is referred to as an HLA haplotype. The probability that siblings will share the same two HLA haplotype is 25%. The probability that an unrelated donor will share the same two HLA haplotype is very low. It's very important in bone marrow transplantation. It's very important for bone marrow transplantation. When a person wants to give achieve the transplantation, get the transplantation from the sibling, the probability of HLA compatibility, full match compatibility, it's more. It's about 25%. But if if the patient does not have any siblings, we need to find a person from unrelated donor, and the probability of full match and compatibility is very low in the unrelated donors. Okay, we have here class 1 MHC molecules and class 2 MHC molecules here. The HLA component in the chromosomes for class 1 MHC molecules are HLA A, B, C, and we have HLA DR, DQ, and DP here for class 2 MHC molecules. It's the structures of the MHC molecules here. In class 1 MHC molecules, which we have in all of nucleated body cells here, we can see an alpha chain here which binds to the peptide, and we have a part of beta-2 microglobulin here. Okay. But in class 2 MHC molecules, which present in antigen presenting cell, for example, dendritic cell, B cells, and macrophages, here we have an alpha chain and we have a beta chain. We do not have any beta-2 microglobulin chain here. Beta-2 microglobulin only seen in class 1 MHC molecules. And the peptide attached to both alpha chain and beta chain here in MHC2 class molecules. We want to, here we show the difference between HLA and MHC. HLA is a gene complex encoding the MHC protein, and MHC a set of cell surface proteins. So the gene is HLA, named HLA, and the protein named MHC. The difference between class 1 and class 2 MHC here, class 1 MHC bind to CD8 positive T cell, class 2 MHC bind to CD4 T cells, and it's sensitive to cytosolic protease here, intracellular pathogens, and class 2 MHC are sensitive to endosome and lysosome proteases. And peptide binding site in cells is reticulum endoplasmic or MHC plasma, and vesicle specialized compartment here in MHC class 2. It's about the peptide size here, and expression and presentation. MHC class 1 expressed by hematopoietic cell presented by all nucleated cells. But MHC class 2 expressed by hematopoietic cells and stromal cell and thymus presented in macrophages, dendritic cell, and B cell. Aren't you tired?
Yes.